Hello, everybody. Let's get this show started. Good afternoon, folks. Welcome to the Lunchtime Discovery Series. It is good to be with you for another edition of the program. It's always good to be with you on our Wednesday afternoon chats. This series is brought to you by the folks at the Office of Environmental Education within the Department of Environmental Quality and us here at the North Carolina Museum of Natural Sciences. I'm your host. My name is Chris Smith. I work here at the museum as an educator and your host every Wednesday. It's fun to host this show because I always get to meet interesting people, and then we get to have great conversations about science and nature and education, all things that I like, and I think all things that you folks like as well. So we all just get to gather here and hang out. Although today I feel like everybody should just get out their gone fishing sign, just hang it right there off of their desk, because that's what we're doing today on the Lunchtime Discovery Fish series. We're going fishing, at least sort of. I think we're going to meet some interesting fishes. We're going to hear some interesting stories and learn about the exceptional place that is North Carolina with today's guest. So let me introduce today's very special guest, Dr. Dan MacGuigan. Dan is the National Science Foundation Postdoctoral Fellow at the State University of New York at Buffalo, and he has spent some time investigating the fish species of North Carolina. Has, I'm sure, going to be some great stories. Welcome to the show, Dan.
Thanks for the introduction, Chris. Let me get my slides up here. It's a pleasure to be with everybody here this afternoon or this morning, wherever you are. And yeah, today we're going to talk a little bit about some of North Carolina's really incredible fish biodiversity. Okay. So like Chris said, I'm Dan. I'm a NSF postdoctoral fellow. And yeah, today we're going to talk about some stories, some biodiversity. The talk's going to sort of be broken down into maybe three-ish sections. First, we're going to talk broadly about the biodiversity of fishes around the world and here in North Carolina. We'll then talk a little bit about what it means to do natural history research in the 21st century. And finally, we'll finish off, or I'll finish off by sharing some of my own research on fishes of North America and North Carolina. And if you're really here, you're really excited to learn more about Lake Waccamaw, you'll have to stick with me till the very end. I promise we will get to Lake Waccamaw at the end of the talk. All right, so one of the things that really stood out, I guess, in my mind, one of the basic scientific facts that I remember learning in high school biology class was that we don't know how many species there are on the planet Earth. There's no comprehensive catalog anywhere that lists every single species that's found here on the planet. And that's pretty remarkable, given all the other scientific advancements that have happened over the past few centuries. We're still missing some very basic knowledge about biology. And when I first learned this fact, I think my thinking was, well, most of those species must be hiding in remote parts of the world, places that are difficult to access for scientists, things like the deep oceans or interiors of tropical rainforests, and there must just be all the species hiding there. But as it turns out, new species are hiding everywhere in plain sight, often in our very own backyards. So take, for example, Europe. Europe is the birthplace of modern Western scientific theory. It's the birthplace of the species as a scientific concept. And yet, even in Europe, this very well-studied continent, we don't know how many species there are. In 1850, there were about 30,000 described species in Europe. Fast forward to the present day, and that number is now around 120,000 species. And that number continues to grow. In fact, the rate of species discovery in Europe is faster today than it ever has been in the past. It's pretty remarkable that even the most well-studied parts of our planet, like Europe or North America, or places that have been the focus of centuries of natural history research, are still hotbeds of species discovery. And this is a bit concerning because, as I'm sure many of you are aware, we are currently in the midst of a biodiversity crisis. It's so bad, in fact, that some scientists have taken to calling it the sixth great mass extinction, sort of linking it with previous, geologic mass extinction events like the end Cretaceous mass extinction event that wiped out the dinosaurs. And this is based on the observation of the number of species that have gone extinct, the rate at which they're currently going extinct, and the sort of number of species that are threatened with extinction around the globe. So this nice little infographic from the New York Times, for instance, shows that about 13% of bird species, 24% of mammal species, and 41% of amphibian species on the planet are at risk of extinction or are threatened with severe population decline. This is a very nice infographic, but it actually only represents about half of all vertebrate species on the planet, because the other half of vertebrate species diversity are fishes. And indeed, fishes diversity globally is also under threat, just like mammals, birds and amphibians. If you're interested in learning more about fish diversity, specifically freshwater fish diversity, I'd highly recommend you check out this nice report from the WWF and a whole bunch of other conservation-oriented groups that was just put out a year or two ago, and talks about the world's forgotten fishes, referring to freshwater fish. Most of us think about marine species as being a threat, the coral reefs being damaged, but in fact, freshwater fishes are also equally at risk. About half of all, or actually a little more than half of all fish diversity on the planet is found in fresh water, which is really remarkable when you consider that only 1% of all aquatic habitats globally are freshwater. So we have 50% of the fish species on the planet jam-packed into just 1% of the available aquatic habitat. And this seems like a potential recipe for disaster, for conservation disaster, because as we know, habitat destruction is one of the main drivers of species extinction. And indeed, 30% of freshwater fishes are considered at risk of extinction based on this international organization called the IUCN's Red List, which classifies species whether as to their status, whether they're the least concerned, they're doing just fine, or they're critically endangered, they're in dire need of help. This is further complicated by the fact that freshwater fish diversity is not evenly distributed around the globe. There are regions of the world that are hotspots of freshwater fish diversity. As you can see on this map here, these areas in dark red or orange are the places where we find the most fish species. And most of those hotspots are located along the equator in the tropics, in the Amazon basin, in Central Africa, Southeast Asia. These sorts of places are home to an incredible diversity of species. But there's actually one more major freshwater fish hotspot, and that's right here in North America, in the southeastern United States, where we have around 1,100 total described species in North America, with a lot of that diversity concentrated in a relatively small portion of the continent. As you can see in this more detailed map, much of that diversity, the red areas of the map, are located in sort of the Appalachians or the Central Highlands, the Ozarks and the Ouachita Mountains. And North Carolina is no slouch here. The western half of the state in particular, where it sort of butts into the Blue Ridge, is one of the most species-rich places in North America in terms of freshwater fish diversity. And even the eastern half of the state, the Piedmont and the coastal plain, are no slouches either. They're colored yellow on the map here, which means they have somewhere in the hundreds of fish species that you can find in these waterways. So if you're interested in learning more about the freshwater fishes of North Carolina, their diversity, which species might be at risk, there are a lot of amazing resources out there. And I'd just like to point out a few of them. Maybe we can put these up in the chat or in the comments of the video later, some of the links to these, or you can pause the video and grab these links yourself. There's a lot of great websites and documentation out there about North Carolina's fish diversity. ncfishes.com has these amazing species pages for every marine and freshwater fish found in the state. If you're looking for a more detailed read, you could check out the Annotated Atlas of Freshwater Fishes of North Carolina, which has been put together by folks here at the museum and at NOAA, and a lot of other just great resources to explore the diversity of fishes as well as their aquatic habitats in North Carolina and in the United States. So let's get into it. Let's explore some of North Carolina's fish diversity. If you're an angler in the room, or in the virtual room, I suppose, you've probably, I'm sure, encountered a lot of these species, these game fishes, these sport fishes that folks like to catch. They're fun to catch, they're good to eat, and they're quite common across the state, right? But even if you spent your whole life with a rod and reel going around North Carolina catching fish, you likely would still only see a very small fraction of the total diversity of freshwater fishes that you can find in the rivers and streams of the state. And that's because most of the diversity of freshwater fishes in North Carolina are non-game fishes. Oh, I should point out here in the previous slide and this slide, all of these photos are, all these beautiful fish photos are from ncfishes.com and used graciously with permission from that website. You should definitely go check them out if you haven't heard of them already. There's just really fantastic, probably one of the best online resources for fish diversity, I think, of any state, or at least very up there in the top of the list. So most of the freshwater diversity of North Carolina are these non-game fishes, and a lot of them are incredibly beautiful. We have things like the saffron shiner, the mountain red-bellied dace. One of my personal favorites, the rainwater killifish. These brightly colored things that look like they belong out on a coral reef somewhere, not swimming around in a little stream. There's weird fish, weird diversity, like the mad toms, like the orange fin mad tom up here, which are actually fishes that produce venom. And if you pick them up in just the wrong way, you might get pricked by one of their pectoral fin spines and find yourself feeling a sting, kind of like a bee. There's oddball fish like mountain brook lamprey and other lampreys. These are jawless vertebrates. They have a circular oral disc instead of a jaw, and they use it, some of them species use it as a sort of means of latching on to other fish as a parasite, kind of like a bit like a little leech might do. And then there's other incredibly gorgeous fish like the Carolina pygmy sunfish. This little fellow only gets to be about 1 inch in length, maximum size, and they're found in these really swampy, hard to access parts of the state. But they're probably, I think, one of the most striking looking little fish that you can find out there. Sorry about the Zoom meeting controls. Hopefully that wasn't too distracting. I don't know why they're popping up. All right, looks like they're good now. You might have noticed, though, on that previous slide, if you're familiar with the fish diversity of North America, that there was one major group of fishes that was missing, and those are the darters. And I've kept them separate because darters have been a focus of my own research for close to a decade now. Darters are this incredibly diverse group of little fishes. It's about 250 species of darters scattered all across North America, or well, pretty much Eastern North America. As you can see from this video, a lot of them spend their time darting around the bottom of streams and rivers, and that's because most species of darters don't have a swim bladder. They lack this organ that allows other fish like that little minnow you could see there to maintain buoyancy and swim around in the water column. So most darters spend a lot of their time just sort of hiding down at the bottom of the streams, and you would never, often you sort of camouflage nicely with the rocks, difficult to sort of notice. A lot of darters are strikingly beautiful, like this Persina here, really gorgeous coloration. In particular, the males will get these fired up breeding coloration in order to attract mates and also to sort of fight off competitors. Here you can see actually a male in the back trying to court a little female darter, or I believe 2 males here sort of squaring off and defending their turf. So an incredibly diverse set of little organisms, and unique to North America, darters are. Let's see. Sorry, I'm having some trouble getting this to go to the next slide here. There we go. All right, so North Carolina is home not to 250 species of darters, but to around 37. And a lot of these darters are, incredibly gorgeous little fish, like the gilt darter, Persina vitis, the greenfin darter, Northnodus chlorobranchium. There's lots of sort of strange-looking darters, like the glassy darter. It's called such because it's sort of, it's kind of somewhat translucent. You can actually see right through to its internal organs under the right light. or things with really weird craniofacial morphology, like the sharp-nosed darter, which has, as the name suggests, a really sharp, pointy nose that it uses to root around and flip rocks. Or the opposite end of the spectrum, things like the snub-nosed darter and their close relatives, which are, I think of them as sort of the bulldogs of the darter world. They have really smushed in snouts and big bulging eyes that really, they do look like the bulldog equivalent of a fish. So a lot of diversity. We could spend, the next half hour just talking about the diversity of darters alone. But we're not going to do that. Instead, we're going to focus in just on one particular darter, or rather, I should say, a complex of darter species called the Johnny darter. And for the next few minutes or so, we're going to try to answer a pretty basic question. That is, how many species are there in the Johnny darter species complex? What I mean by a species complex is a group of species that are all closely related and difficult to distinguish from one another. They might be morphologically similar. The taxonomy might be quite confusing. And indeed, that's the case for the Johnny darter. This little fish was first described by the ichthyologist Constantine Samuel Rafinesque back in 1820. And since then, there have been a whole host of other names used to describe species in the Johnny darter complex, species or subspecies, and it's been just a kind of a confusing mess for the greater part of a couple centuries. So in my research, actually during my PhD, I tried to tackle this seemingly simple question of how many species there are in this group. So just to introduce you to the two main players in this group, there's currently 4 species recognized within the Johnny darter species complex, but the two ones that are sort of the I guess, most important for this talk are the Johnny darter, Ethiostoma nigrum, and the tessellated darter, Ethiostoma olmstedii. These two species have pretty massive ranges. The Johnny darter in particular has one of the largest ranges of any darter species, all the way from the lower Mississippi out near the Gulf Coast to the Hudson Bay up in Canada. And the tessellated darter is no slouch either. Its range spans most of the Atlantic seaboard from all the way up from New Hampshire down to Florida. There's a little isolated population you could just barely make out right there. And there's some other oddities about these two species. They have a large contact zone, which I've shown here in yellow on the map, where the two species overlap and can be found in the same rivers. There's also this odd little pocket of green on the map here. That's not just a little coloring mistake. Currently, there are supposedly Johnny darters actually living east of the Appalachian Mountains in this little isolated pocket surrounded by tessellated darters. So how do we actually go about studying this complicated species complex? Well, let me walk you through what the process, or how I would sort of define the process of natural history research in the 21st century. But let's first start by contrasting that with the origins of natural history research back in the 1800s. So folks like Rafinesque here would go around in their little horse-drawn carriages to the wilderness, of North America. And at the time, that was places like Ohio. You'd take detailed notes of all the species you encounter, often, with beautiful little illustrations. And you bring back those notes as well as some specimens to your museum or your university, where you'd write up a description of everything you saw on this big collecting trip, including any new species you might have come across. So, for here in 1820, Rafinesque for the first time describes the Johnny darter, Ethiostoma nigra, he calls it. So what does this process look like in the 21st century? Well, it's actually the sort of backbone of this work is remarkably similar to how it worked in 2 centuries ago. We go out to the field, we take detailed notes of all the fish species we see and all the sites that we visited. We collect specimens, we take photographs. I'm not a very good illustrator, but my phone is full of fish photos from all these field trips. And we bring all that data along with some specimens back to our universities and our museums. And once we're there, we describe, everything we saw. We write up papers. And if we happen to find any new species, we'll do some analyses and we'll write up a description of the new species, like you can see here, for a new species called the Etowah bridled darter, I should say. So a lot of similarities over the course of 2 centuries. A lot of things have remained the same about how we do natural history research. But of course, there are a lot of differences. So let me walk you through the whole sort of process, at least my process of how I go about doing this kind of research. First off, we have to start by identifying sampling sites where our species of interest might be found. And here's where we have a leg up on folks like Rafinesque. There are a number of amazing databases out there that exist for historic records and contemporary records of all of these amazing fishes that I was showing you a few slides ago. Places like websites or repositories like GBIF and VertNet, and things like iNaturalist, which many of you may have used. It's a little app you can download. You can it's a sort of citizen science-based approach where you can identify species you see in the field and share them with others, sort of social media for natural history, right? And if you're using that app, I'd like to say thank you, because we scientists actually do use that information to try to locate, sites or places where we might be able to find the species we're interested in researching. There's also, of course, amazing books like the Atlas of Freshwater Fishes in North Carolina or New York that we can use, where they compile, decades worth of work and important information about the species we might be trying to find. Another thing that's different from how Rafinesque and his fellows operated, his fellow natural historians operated in the 1800s, is that we have rigorous permitting systems that we go through in order to make sure we're performing this research in an ethical manner, in a way that minimizes damage, harm to the fish and to the populations that we are surveying or sampling. So we work with state agencies like the North Carolina Wildlife Resources Commission. We also work with federal agencies sometimes, as well as these internal committees like the Institutional Animal Care and Use Committee. All of this to make sure that we're doing the best that we can in terms of the ethics of our research. Once we've done all that sort of background work, then it's time for the fun stuff. We actually get in our truck or whatever, and we drive out to those field sites. And again, this is where we have a massive advantage over the 1800s, where they're relying on very limited infrastructure, poor roads, and can only visit some small segments of rivers at a time. We're able to drive, for instance, cross country practically in a matter of days. This was actually the very first collecting trip I ever did, sampling trip I ever did during my PhD. We drove from Connecticut all the way down to New Mexico in the span of just 2 days. We search for suitable habitats, and then we collect. We find fish and other critters that we're looking for. And a lot of the collection techniques that we use are similar to those used by 18th century or 19th century natural historians, things like seine nets. But we do have some fun gadgets that help us more effectively sample, things like backpack electroshocking units that we can carry around and look as we're looking for fish. Then comes perhaps the most challenging part of this whole endeavor. We have to identify every fish we catch. I'm showing you here 4 different photographs. These are actually 4 different darter species. And some of these species are found in the same rivers and streams. As you can imagine, if you're standing there, in your waders, covered in sweat and grime, looking at a little fish in your hand that's only a couple inches long, it can be really challenging to tell apart these different species. But it's critical that we do this well, because first of all, we need to make sure we're catching the species we want to study. Second of all, we need to make sure we're not catching any species that might be of conservation concern. So it's actually a big part of our training as natural historians, learning how to identify species. Once we've collected the species we are looking for, we will take some specimens, we'll preserve those specimens and catalog them, usually on, maybe on the bed of a pickup truck or late at night at a campsite here. And this is how we use a lot of the same techniques here that folks were using over the past 2 centuries. And this is crucial. This part of the process is critical, because by preserving these specimens, we're ensuring that they can be used for many, many generations of future researchers. This little jar of fishes here I'm showing you is from the Cornell Natural History Museum. I was up there a few years ago looking at some fish, trying to take some photographs for some of my own research. And these fish were collected in the 1930s, almost 100 years ago. And yet they're in really, solid condition, good enough that I can use them for my own research purposes. So by preserving and cataloging all these specimens we find, we're able to ensure that many, many future researchers will be able to benefit from our work. And as just a brief aside, if you've seen other videos in this series, you probably already know this, but natural history museums are far more than meets the eye. Their public exhibits inspire us, and they're certainly fun to visit. But the collections that are behind those exhibits are the really important resource for biologists like myself who want to study natural history. The last thing we do that folks in the 1800s would never have thought of is we take tissue samples to preserve DNA. Structure of DNA was only discovered in the 1950s, so it's a relatively recent development in natural history research, taking tissue samples for preserving DNA. So each of those little tubes in this little container up here contains a small piece of tissue from a different fish, which we can then go and analyze, extract their DNA and sequence it. And these tissues are also preserved just like the fish specimens so that future researchers can benefit from the work that we've done. Okay, let's get back to the darters here. We went out and sampled 179 unique localities across the range of the Johnny Darter species complex. You can see all the spots where we sampled are these little white points scattered all over the map. And we collected 444 tissue samples from fishes across the range. And as you might guess, this is not something I could do on my own. There were a ton of folks who were involved in this research, in this sampling effort. People who worked directly with me in the field, of course, but many other folks at other institutions and agencies, including here at the North Carolina Museum and with the DEQ, who've contributed to this project. So thank you to all of those folks who've put in hard work to help us understand this little species complex of darters. We take these tissue samples and we go and sequence the DNA. From this DNA, we can learn a lot about how all of these different fishes are interrelated. Just as a brief reminder, we've understand that the general structure of a genome works something like this. We have individual molecules, A's, T's, G's, and C's, that are strung together in these double helix structures into long strands of DNA. The DNA contains genes which can encode for all the proteins that are the instructions for how we make cells, how we make ourselves and organisms, and it's all packed nicely into these little chromosomes. But how do we actually get the information out of the genome? Well, the way in which we did this for this particular project is a technique called restriction site associated DNA sequencing, which is a bit of a mouthful, but it's a really simple idea. We could sequence the whole genomes of 444 fishes. That's possible, but it's quite costly, both in terms of time and money to actually do that kind of work. So rather than sequence the whole genomes, we decided to sequence segments of the genome. We first chop up the genome into little bits using these restriction enzymes that recognize specific sets of sequences, specific DNA, and they'll cut the DNA at that site. Once we've cut up the DNA into a bunch of smaller fragments, we isolate a subset of those fragments. And now we have a smaller portion of the genome. We're still getting a lot of information here, but not the entire genome. And we take that subset of fragments and we sequence them, in this case, using a technology called Illumina sequencing, which is really cool in its own right. It uses fluorescence to actually detect whether an A, a T, or C, or a G is present along a strand of DNA. And these machines are actually really quite large. I didn't realize this until just recently. Some of this is sort of their latest sequencing platform. It's almost as tall as these two. Different. We can learn about the evolutionary history of these fishes. We reconstruct the evolutionary history using what we call a phylogenetic tree. In a phylogenetic tree, in this particular one, we have the past on the left and the present on the right. So all of the tips of the tree represent living species or living individuals or populations. And if we trace those back until they all converge, we find the most recent common ancestor of all of the living species or individuals that we have sampled in our data set. This phylogenetic tree is analogous to like a family genealogy. we're trying to trace back how you're related to your third cousin twice removed or whatever, you can follow your family's genealogy. Well, just like you do that, we can follow the evolutionary history, the genealogical history of these fishes and try to understand how they're related to one another. So to jump right to the results, this is the phylogenetic tree on the left for the Johnny darter species complex. There are 444 individual little tips at the right side of this tree. If I was to zoom in, you'd be able to see each of them is labeled with some information about the species, where it's from, all that stuff. If we step back and look at sort of the broader picture of this tree, we can see that there's something interesting going on here. There are actually two different distinct groups that make up the tessellated darter in this phylogenetic tree, and they're not each other's closest relatives. The Johnny darter is actually more closely related to some of the tessellated darters than those tessellated darters are to each other. So we sort of have the tessellated darter is this broken up, what we call a paraphyletic species. And that's something that really we don't want. We don't want to be describing species that are not sort of each other's closest, that don't sort of form a single unit, a single group on the phylogenetic tree. So the simplest thing to do here is, well, maybe one of these species, one of these tessellated darter groups should actually be considered a distinct species. And as it turns out, if you look at where these two different groups are located in geographic space, they break down very nicely with the dividing line right here in North Carolina. So this pink group at the bottom is sort of makes up the southern half of the range of the tessellated darter, and the orange group in the tree at the top makes up the northern half of the range of the tessellated darter. So this is quite exciting. Using these genetic techniques, we've discovered, or we found that there's a new species sort of hidden within the Johnny darter complex. And it turns out there is actually a name, a scientific name that we can use that someone described more than a century ago called Ethiostoma maculataceps. And so we have a brand new species here, the Ethiostoma maculataceps. But what do we call it in terms of the common name? This is just something sort of an interesting little aside. There's a lot of guidelines for how we use scientific names. There's very clear-cut rules about which name gets priority over what name. So that's easy enough. But there aren't really any such guidelines about common names of species, which is a bit of a shame because, colloquially, we all refer to species by their common names. It's really a much more useful in some ways name for a species when just talking with any old person. So there are some ideas out there that's been called the southern tessellated darter, the spotted head darter. My personal favorite, though, and the one I'm going to use for the rest of the talk, is the two-spine darter. And that's because one of the characteristics that distinguishes this new species from all the other species in the Johnny darter complex is that it has two spines in its anal fin. All the other species have just one spine in their anal fins. The story, though, is actually far more complicated than just these three species. There are a whole host of other lineages we identified in these analyses and in this paper that likely merit recognition as new species. I've highlighted them on the map here on the right with these numbers. Unfortunately, I don't have time to go into all of the sort of craziness of this species complex today. But I just wanted to point out that there are quite a few new lineages that we've discovered with these genetic analyses. But I will take some time for the remaining 10 or so minutes of the talk to show you a little bit about one of these species, and that is the Waccamaw darter. We're finally here. We're finally at Lake Waccamaw, like I promised. The Waccamaw darter is, I think, one of the most unusual darter species out of all the darter species. And that's because it's found only in a single lake in North Carolina, Lake Waccamaw. It's found nowhere else in the world. And this is a really quite small lake. If you've ever been there, you could paddle across it in just a couple hours. It's not a very large body of water. And Lake Waccamaw and the Waccamaw darter are nested within the range of the two-spine darter, that new species I was just talking about, on the previous slide. And so, really interesting contrast here. We have a species, the Waccamaw darter, that's found in only a single lake, and then a species, the two-spine darter, that's spread across 4 different states. And it's actually, in the scientific literature, there's been quite a bit of debate about whether the Waccamaw darter is even really a distinct species from the two-spine darter. Maybe it's just sort of a unique population that's found in Lake Waccamaw and not a distinct species. The problem is nobody's ever really done a detailed look at taken a detailed look at the genetic makeup of these two species to try to answer that question. So we went ahead and tried to do this. We sampled 13 sites, 4 or 5 of them in Lake Waccamaw, the remainder in the Waccamaw River and the adjacent Black River. We collected two-spine darters and Waccamaw darters from all of these locations. And we used the same DNA sequencing technology that I showed you on the previous, for the previous sort of research project on the Johnny darter complex. We used the same approach to sequence the DNA of about 100 fish from all of these sites. And what we found is that the Waccamaw darter is a very young species. This figure here is showing you the phylogenetic tree, if you will, of the Waccamaw darter. It looks a little bit different than that phylogeny I showed you before, but it's the same idea. At the top of this, we have the past, and at the bottom of this figure, we have the present. Our analyses show that the Waccamaw darter in the lake split off from the two-spine darter in the Waccamaw River about 11 to 45,000 years ago. And this is a very recent event in terms of speciation. Most darter species are on the order of millions of years old, not tens of thousands of years old. But what's cool about this is that our estimate of the age of the Waccamaw darter is very consistent with the geologic estimate of the age of Lake Waccamaw. This is based on independent evidence from sediment core analyses. Folks think that Lake Waccamaw originated 15 to 30,000 years ago, which is around the same time when the Waccamaw darter first came into being. And this is very comforting because it would be a very sort of strange result if, for instance, the Waccamaw darter was way older than the lake itself. That wouldn't make a lot of sense. So what we see is that these two species split off from one another and were sort of functioning as their own, doing their own thing for 10,000 years or so. But in more recent times, in the past few hundred or few thousand years, the two species have started to hybridize and produce individuals that are intermediate in their genetic ancestry. So this is a bit odd. Why is there this separation between these two species? Why weren't they producing hybrids this entire time? The Waccamaw darter is found in the lake, and the two-spine darter is found in the river right next to the lake. You'd think that they would be able to sort of intermingle constantly throughout their history. But we think that this might not have been the case because Lake Waccamaw may have been historically isolated from the Waccamaw River. There's some scientific evidence to back this up, but I'll share with you instead an anecdote that I think is kind of interesting. I happened to be perusing Facebook one day, procrastinating from some important project or whatever, and I came across this post from the Lake Waccamaw Depot Museum, which is a small history museum located in the town of Lake Waccamaw. And they had posted they post a lot of old photos and newspaper clippings about their town and about the lake. And this one particular clipping caught my eye, so I reached out, got in touch with some folks there who put me in touch with people at the News Reporter, the newspaper where this article originated. And we were able to track down the original article from 1940. And what really caught my eye about this was the headline that says, "Drive auto around lakes for the first time in 15 years." In 1940, there was a pretty severe drought where the lake dried up enough that folks were actually able to drive their 1940s automobiles around the entire bed of the lake. Their lake was entirely cut off from the river. And you can see just how dramatic this is if you look at the little pier, the little jetty here on the left side of the screen. That's the normal water levels of the lake. And it's not just this one time. As you'll notice in the title, it says first time in 15 years. So these drought conditions had happened previously, 15 or so years ago. Why isn't this still happening today? Lake Waccamaw is not drying up like this anymore. Well, the reason is there's a small dam that's been built that actually regulates the water level in the lake and keeps it high year-round, even during the driest months of the summer. And that dam actually counterintuitively maintains the connection, therefore, between the lake and the river, because the lake never dries up like it used to. But prior to that dam, for thousands of years, Lake Waccamaw may have undergone these periodic fluctuations where it dried up and lost its connection to the Waccamaw River. So we hypothesized that during these periods where the lake dried up and was isolated from the river, that was when the Waccamaw darter was first able to start diverging from its counterpart, the two-spine darter, in the river. And it did so without having to worry about any of that messiness of hybridization that might sort of break down the barriers between the two species as they're starting to form. Okay. So that's a little bit about the evolutionary history of these two species. But as you might guess, the lake and the river are very different environmentally. First of all, they're different just in terms of their scale, their size. The Waccamaw River is about 140 miles long. Along its course, it's variable in its depth. There's lots of different substrate types and lots of different habitat structures that these species can utilize. The lake, however, as we've already established, is pretty small. It's only 14 square miles, and it's very shallow, with a maximum depth of only about 11 feet. The lake environment is pretty homogeneous. Most of the substrate is just sand, and it's open. There's very little sort of cover or structure for these species to sort of hide in from predators. These water bodies also differ in their water chemistry. The river is a black water river. It's got lots of tannins, it's low alkalinity and low pH. The lake, on the other hand, is actually a clear water lake, and that's because there's a big limestone deposit that maintains high alkalinity and keeps that pH at a sort of neutral level. So given all of these environmental differences between the habitats being occupied by these two species, we wanted to know, are there accompanying differences in their ecology and their morphology? So working together with Dylan Wainwright at Purdue University and Rich Harrington, who's now at the South Carolina DNR, we were able to do a bunch of work to show that, yes, indeed, the lake and river darters are morphologically and ecologically distinct from one another. To look at the morphology, we actually did micro CT scanning of these little tiny fishes that are only a few inches in length, and we're able to make 3D models of their skeletons from this. We then measured a bunch of different traits, particularly focusing on traits like the jaw shape, jaw lever ratios. So for instance, we looked at this ratio called the jaw opening ratio and looked at it for a number of fish from both the lake and the river. Each little point on this plot shows an individual fish, and they're colored by whether they're from the lake or the river. And as you can see, fishes from the lake, the black points, tend to have lower jaw opening ratios than fishes from the river. What's really cool is that this ratio, this jaw opening ratio, is strongly associated with the foraging behavior of these fishes. Lower jaw opening ratios are associated with more suction feeding. And this matches up with what we find when we look at the stomach contents of these fish. We found that Waccamaw darters tend to eat more amphipods and ostracods, these free-swimming crustaceans that would require some sort of suction in order for these fish to sort of chase them down and feed on them. In contrast, the two-spine darter in the river, we found their stomach contents contain far more ephemeropterans, trichopterans, and isopods, mayflies, caddisfly larvae, and sort of pill bugs or roly-polies. And these little bottom-dwelling organisms have much tougher carapaces that would require a lot more biting force in order to eat. So it's really neat. We found that there's this link between the morphology and the ecology of these fishes that may have been important in initiating the speciation between these two lineages. Lastly, and very briefly, I'll just say we have undertaken to sequence the entire genome of the Waccamaw darter. This is more recent work done partially during my postdoc here at the University of Buffalo with Trevor Krabenhoft and grad student Nate Backenstoes. We use this technology called nanopore sequencing, where the DNA is, as the name suggests, forced through this tiny, tiny little pore. And by looking at changes in electrical signal as the DNA goes through the pore, we can tell the sequence of A's, T's, G's, and C's that make up that DNA molecule. And what we get out at the end is kind of a little bit boring looking, but it's a series of those four molecules, T, C, G, and A. Using this genome, we can look for regions of the genome that have a lot of genetic differences between the lake and the river darters. In this plot, I'm showing you a metric called FST. If FST is high, that means there's lots of genetic differences. If FST is low, that means there's really no genetic differences. And when we look across all 24 chromosomes of the darter, we find that there's one region of the genome in particular that stands out, right here on chromosome 9. There are a lot of sequences here that exhibit lots of genetic differences between the Waccamaw darter and the two-spine darter. Here, if I zoom in, you can kind of see this more clearly, just looking at chromosome 9. The sort of bottom, the latter bit of it has this elevated genetic differences between the two species. What might actually be causing this? Why is it just this one region of the genome where we see dramatic genetic differences between the two species? One possible explanation is something we call a chromosomal inversion. This happens when chromosomes are being duplicated. The part of the chromosome can actually break off and reverse in orientation, and it's then reattached, but in the opposite direction. This is problematic. If you remember back from high school biology class, there are lots of times where chromosomes need to pair up, such as during meiosis or mitosis. And if you've got this chromosomal inversion, all of a sudden now your two chromosomes in the pair cannot properly match up to one another because they have one of them in the sort of reverse orientation. And this creates all sorts of issues, for an organism. But in the case of the Waccamaw darter, we think, or we have evidence that a chromosomal inversion is actually why they are genetically different in this region. When we look at two different genomes on here, I'm showing on the right the Waccamaw darter chromosome 9 as the x-axis and the Johnny darter chromosome 9 as the y-axis. And when we compare how those two chromosomes line up to each other, we see that most of their chromosomes line up sort of in this one-to-one orientation where they're both sort of, the two sequences pair up nicely with each other. But the latter half of the chromosome, we see that one of the DNA strands is reversed in orientation, and that is a chromosomal inversion. So only this chromosomal inversion really on chromosome 9 of the darters is where we see these massive genetic differences. And what's really cool is if we actually look at what specific genes are found within this inversion. There are about 400 genes found in this region of the genome, and a number of them we noticed are involved in development of the retina or development of the eye. Why might this matter for these fishes? Well, as I mentioned a few slides ago, the lake and the river have different sort of water conditions. In fact, the lake is a clear water habitat. It's open, there's no cover, and lots of light penetrating through to the bottom of the lake. The river, on the other hand, often has very closed canopy. It's lots of shade, right? And it's a blackwater river, so the clarity of the water itself is not very high compared to the lake. So it makes a lot of sense that these two species would need to have sort of different developmental strategies for their eyes in order to be adapted for living in these different environmental conditions. We think this is evidence that the Waccamaw darter has what's called a supergene, which is a set of genes that are located close together on a chromosome that work together to produce some complex trait, such as an eye. I'm going to skip through to the end just so we have time for questions and say that the Waccamaw darter is only one of 3 fish species that's found that's unique to Lake Waccamaw. There are actually 2 other species of fish, the Waccamaw killifish and the Waccamaw silverside, that are also found only in Lake Waccamaw and more or less nowhere else in the world. And we've started to dig into these other 2 species recently to look at whether there's any evidence of repeated or convergent genetic evolution between these 3 species of fish found in Lake Waccamaw. With that, I'll just kind of go back to this, these beautiful photos of this fish diversity from North Carolina, and just to say that each of these species shown here has its own story to tell. Some of them might contain undescribed species, or others might have sort of secrets to their genome that we have yet to unlock, like we saw with the Waccamaw darter. And it's really important, more so now than ever, that we try to protect this species diversity so that we can learn about these evolutionary and ecological stories from all these different incredible fish species. with that, I've already thanked the folks who helped out with the specimen collection, but there are many other funding agencies and people who have been part of this research over the years. And we have a bit of time for questions. So happy to take any comments or questions you might have about fishes of North Carolina or the Waccamaw darter.
Dan, thank you so much. Excellent stuff.
Thanks, Chris.
Great presentation. Oh man, such cool stuff. One, the fish look cool. Like they just look like neat little animals. But then to learn that there's such great specificity, like right here in North Carolina. And even in places that I don't necessarily consider it. Like if we're talking about diversity of species in North Carolina, we tend to not be talking about the Piedmont or the coastal plain a lot. A lot of scientists and people will talk about the number of fishes or salamanders and amphibians that are up in the mountains. But to know that that we also have great diversity out along our coast and in the coastal plain, that's exciting.
Yeah, the Blue Ridge gets a lot of attention and rightfully so. But there's a lot of cool species everywhere in the state, and a lot of things like you saw with the Waccamaw darter and the other Waccamaw species that you can't find anywhere else, right there on the coastal plain. Kind of a weird spot for them, considering it's been covered by the ocean periodically throughout its history. But
yeah, absolutely, exciting stuff. So I'm going to remind all of our viewers, there's a few questions that have already been dropped into the chat, but we do have several minutes. So if you've got questions or thoughts, let me know, drop them in there, and I'll be turning to you folks in just a moment. But Dan, a question that was coming up from me was that I don't think I realized that you could get new species breaking off and becoming new species as quick as a few thousand or a few tens of thousands of years. Like if Waccamaw is only maybe up to 30,000 years old, and then the darter is right around that age, to me and my limited knowledge of species diversity, that seems like a really quick turnaround for these fish to diversify in that way.
Yeah, it definitely is unusual. I kind of mentioned that briefly too, that if we have age estimates for pretty much all the species of darters as a good example. And if you look at most of those species have split, like, pairs of species split from each other on the order of, 1 to 5 million years ago. Pretty deep time we're talking about here. And for something to be diverging so quickly, in this case, only 30 or 15,000 years it's very unusual for the Waccamaw darter, but it's not unheard of in biology. One of the great examples of this are the cichlids, these little fish you've probably seen at pet stores, in these, they're brightly colored, and they have these massive radiations where there's been like dozens or hundreds of species forming in lakes, often actually in the New World and in the Old World, in Africa and in South America and Central America. And on similar time frames we saw with the Waccamaw darter, just a few thousand or a few tens of thousands of years. And they're even more dramatic because it's like a single lake where we suddenly see dozens of species forming rather than just the one species like we saw with the Waccamaw darter. So in some senses, it's a really unusual occurrence. But when you kind of consider, fish diversity more broadly than just more than just darters, it's not all that uncommon.
That's fascinating. Thank you. All right, some questions from our viewers for you. Ready? All right. Ashley wrote, what led you to the initial discovery that Olmstedii was a paraphyletic species?
Yeah, so there's been some previous work done on this species complex, the Johnny darter species complex. So we had a few clues. One of them was actually from back in the 1950s. This guy named Charles Cole did his PhD dissertation. He's kind of like I guess he was my predecessor in spirit, although we have no direct connection and I have never met the man. Don't know what he did after his PhD. But he was one of the people who first really dug into this complex, and just looking at morphology alone, like how the fish look, he realized there's all this weird complexity in the Johnny darter species complex. Like he was actually, at first he was thinking that pink species, Maculiticeps, that we split off, he's one of the first people to kind of realize, hey, there's something weird going on down here in the south. the southern portion of its range, these fish look different. He also noticed a lot of the other diversity of these fishes. So, even dating back to 60, 70 years ago, people realized there's some weirdness in the group. For the actual paraphyletic part of it, the phylogeny, that was, there's been one other study that showed some strangeness with the phylogeny of the darters about 10, 15 years ago now, something like that. But there hadn't been any really sort of big genomic assessment of the species phylogeny until I started working on this a few years ago. So the way we discovered it is partially just a new result, but it's also something we weren't totally surprised by because of this long history of natural history research that had been done on the species complex.
Yeah, a question that came in right after that one from Miriam, I think you kind of just answered it, was getting at the differences between darters with one spine, darters with two spines, and knowing that the two-spine darter was limited to this more southern range.
Yeah, so the morphological side of it, the one spine versus two spine stuff, is pretty well established by that guy named Cole. I don't know if any of his stuff is accessible, freely accessible online, but if you dig deep enough, you might be able to find it. But yeah, he actually went out and measured, collected lots of data about the traits of these fishes, their spines, their scales, all sorts of counts like that. And it turns out if you overlay his data that he collected about the morphology onto the genetic data that we collected about the phylogeny, the two match up pretty well in some cases, particularly with this spine trait. So if you're out there and looking at these fish in the Cape Fear River or the PD River Basin, and you ever catch one of these darters, you take a real close look at the anal spine or the anal fin, you'll likely see that there's two little spines there. If you went just north into say the Roanoke or the New River or the Tar River, those you'd see just the one anal spine there.
Fascinating stuff. So let's see. Next one, Parker wanted to know if any of these species had been kept and maintained in laboratory settings, or are they all done with preserved specimens and doing the work out in the field. And I'll add that there's a viewer, David, who did some of this work on Waccamaw fishes. He wrote back in the 80s out of UNCW with the Lindquist lab.
Yeah. And I know some of that work that was published from the Shoot, JR and Peggy Shoot. That's kind of the foundation. A lot of that stuff is pretty foundational for the work we did here. I didn't have time to go over it, but that early work in the 80s on the Waccamaw darter was pretty inspiring to a lot of the research I've done. And actually, JR, I think, was the one, he actually did do a little bit of early genetics, old school genetics work on this fish, using a technique that has called, I think it was Allozymes or something, looking at the visualizing DNA on a little gel like you kind of see in the movies or whatever. We don't really do that anymore for most of our analyses, but he was finding some of these same differences just with much older technology. To answer the first question, we have not, none of this work is done with fishes in the lab. All of the work I presented here was using tissue samples that were collected from wild populations. But there would be really some cool things we could do if somebody wanted to spend a couple summers down there. It'd be really interesting to see how the Waccamaw darter and the two-spine darter interact behaviorally, whether there's any sort of differences that might help maintain their species integrity. We don't really have a good answer to that as far as I know.
Awesome stuff. Let's see. Do you think that their eye adaptations could be influenced by their diet? Well, I think you indicated, you indicated the mouth shape might be influenced by diet. What about other features?
Yeah, for sure. I mean, vision is important for how these fishes forage. I mean, well, there's kind of two primary components of that. There's olfaction, there's sense of smell, but the vision is pretty important to how these fishes locate their prey as well. again, this would be sort of stuff that you'd have to do some real hard experiments on, with fishes in the lab, to link it back to the previous question. But there is some tantalizing evidence here. There was I'm blanking on their name, but there was a recent, I think it was a master's thesis that came out from a student who was studying the cones and rods in the Waccamaw silverside and the Waccamaw killifish. Sadly, not the Waccamaw darter. I don't know how that one slipped through the cracks, but the other two species that are found in Lake Waccamaw. And comparing their cones and rods to the cones and rods of their closest relatives in the rivers. And they were finding some really striking differences in, I don't remember if it was the number of cones or the shape or something like that. But these sort of visual differences could certainly be very important for how these fishes forage and locate their prey, but likely for a whole host of other reasons too. Avoiding predators and finding mates is another, or two obvious ways in which vision might play a role.
Fun, interesting stuff. All right, I'm over time, but so I'm going to go to one last question, because I like this one in particular. Could similar speciation events have occurred in other Carolina bays like Lake Waccamaw throughout the region?
That is a really great question, and something we've tried a little bit to get at. We actually spent a few field trips going around some of the other lakes, in the other bay lakes in the Carolinas, White Lake, Phelps Lake, and yes and no. There's not a lot of evidence of this yet. Lake Waccamaw certainly seems to be pretty unique among the bay lakes. It's the only one that has this kind of level of endemic diversity that we see. There's also possibly an endemic catfish in the Waccamaw, and as well as some mussels. There's all sorts of weirdness. But some of the other bay lakes do have some possibly indicators that there's been some speciation stories like this. Actually, up in Phelps Lake, in the northeastern part of the state, you can find killifish that right now are considered to be Waccamaw killifish because they look like the Waccamaw killifish. But to the best of my knowledge, there's been no genetic study to show whether or not they actually are the Waccamaw killifish that have been somehow transplanted up there. It's possible that those could have evolved in Phelps Lake independently, right? Evolved to look like the fishes in Lake Waccamaw. So that's a great question, and something we're really interested in pursuing in the near future.
Fascinating stuff. Dan, thanks for being on the program today.
Yeah, thank you so much for having me, and happy to answer any other questions if you have, want to reach out. And I think I don't know if my contact info is available anywhere on any of the postings, but you can find me online.
Yeah, if they can find this YouTube video, they can probably figure out how to get an email off to you as well. And because we did have a few more questions that came in, but I don't want to keep people. We're already over everybody's lunch hour at this point. So, Dan, thanks for being here. Viewers, thanks for tuning in today. This was a really interesting stuff. I can't wait to learn even more about it and do some reading. Lots of great resources and links to resources were posted up in the chat, folks. So get to know your North Carolina fishes. I think it'll be worth your while. We will be back here again next Wednesday for another edition of the Lunchtime Discovery series. Don't miss out. Make sure that you're following the Museum of Natural Sciences and the Office of Environmental Education on social media, where you can get updates about this program and many more that we're all doing, all kinds of great science and nature stuff for you to take in every single day of the week. And you can also sign up for the Lunchtime Discovery Series newsletter. That way you can get the link to this program every week in your inbox, and then you're just a click away from being able to join us and learn something new. So from me and everybody else, thanks. We'll see you again next week. Bye, everybody.